welcome. We're very pleased to have you joining us this morning and really excited to have our friends from Dyne Therapeutics with us to share some really exciting information with you. This presentation will be recorded, so just be aware of that. And if you could just stay on mute, that would help with the sound. But if you have questions, we welcome them in our chat or in our Q&A. You can type them in, and we'll be sure to leave time to respond to those once we get through the presentation. So just in the way of introduction, my name's Lisa McCoy. I'm the CEO for Defeat Duchenne Canada. For those of you who may not know a lot about Defeat Duchenne Canada, Defeat Duchenne Canada is the country's only national charity that's dedicated to ending Duchenne muscular dystrophy. Our goal is always to provide leadership in research, advocacy, and support to ensure that every Canadian affected by Duchenne muscular dystrophy is able to live long and active life. So I've talked about our main areas of focus: education and support. Certainly this is part of our education, our webinar today. We love to make sure that we're able to share up-to-date information with you as we understand how important that is. We are stepping up our efforts all the time in advocacy to make sure that we are your voice with our elected officials and with others who have the opportunity to make policy change and changes in process that will make life better for our families and those who are living with Duchenne muscular dystrophy, and of course our research funding. And to date, since our founding 29 years ago, we funded research, which we're very proud of, and it's all thanks to our donors and our supporters. So we've made great strides, and we have a long way to go, and we will continue to work on that. The other thing I'll just share with you is that just in June last month, we launched our next strategic plan, our 2024 to 2028 strategic plan. Focuses on the pillars that I talked to you about, as well as community engagement. We're always reaching out and doing everything possible to engage as many Duchenne families across Canada as possible with our organization so that we can support one another, share information, and build a strong community for those who are living with Duchenne every day. And in addition to that, we have our enabling strategies, without which we couldn't do any of the work that we do. So fund development, obviously raising money to fund our mission is critically important, and we rely on private individuals, donors, sponsors to help us to do that. We're extremely grateful for their support. We have wonderful teams of volunteers, both our boards of directors, our committees, but also our volunteers all across Canada who do tremendous work for us. And of course, collaboration. As a small national charity, we really rely heavily on partnerships and collaborating with other like-minded organizations, others in the rare disease community, others working in muscular dystrophy and Duchenne muscular dystrophy. Of course, our pharmaceutical partners, our clinicians, our researchers. So collaboration is absolutely key to us in building capacity to be able to make the biggest impact possible. So without further ado, with completing that little intro of Defeat Duchenne Canada, it's my great pleasure to welcome Dr. Dugar today from Dyne Therapeutics. Dr. Dugar is the Chief Medical Affairs Officer. He has over 20 years of experience in clinical and commercial development, as well as real-world evidence, which we all know is extremely important. So at this time, I will turn it over to Dr. Dugar. Thank you so much for being here, and we look forward to hearing your presentation.
Super. Thank you so much, Lisa. Hopefully you can hear me all right. So that was a very nice introduction, Lisa. Thank you so much. As Lisa said, I'm the Chief Medical Affairs Officer at Dyne Therapeutics. First, I want to thank Rick and all of his leadership to defeat Duchenne over the years, and an amazing partner to the company and to me. And so thank you, Rick. I'm sure you're listening. And I'd really like to take this opportunity to thank Lisa, Kate, and the entire Defeat Duchenne organization for the opportunity to present Dyne's Duchenne program. It's always a privilege to speak to the Duchenne community, and I want to thank you for your time and attention today. With me today, not on camera at the moment, is our amazing head of patient advocacy, Catherine Beaverson. As many of you know, Catherine has been part of the neuromuscular advocacy community for over a decade and brings tremendous experience and passion to the role. And so she may be joining me at the end of this, should there be some Q&A that we want to take. Next slide, please. I'd like to remind everyone that we will be making forward-looking statements today, and you can refer to our website for more information. Next slide, please. So this is really important, and I begin with this slide for most of my presentation, because at Dyne, it truly starts with our mission, which is to deliver life-transforming therapies for people living with serious muscle diseases, with Duchenne being a priority. Our mission is woven deeply into our core values and inspires our employees each and every day. Next slide, please. So you've seen me present our platform in the past, and I'll go through it really briefly here in a moment. But I did want to highlight that there are essentially three key challenges as to why first generation exon skipping therapies have significant limitations on dystrophin restoration and functional improvement. The first is what we would talk about as nuclear localization of antisense oligonucleotides like PMOs to be able to harness the cellular machinery to target the genetic basis of disease. So it's really getting that PMO into the nucleus. That's kind of the first challenge. The second is that it's targeted delivery to muscle fibers. So getting it into the key muscles and have targeted delivery with limited or no off-target effects. And the third is broad distribution in those muscles that matter in Duchenne. So not only being able to get into the nucleus and into the muscle fibers, but being able to have broad distribution so that you can produce a large amount of dystrophin and therefore have some functional benefit. So the fourth platform is Dyne's solution to overcoming these historical challenges. The platform is expressly designed to efficiently and reliably deliver antisense oligonucleotides like PMOs to skeletal muscle, cardiac muscle, smooth muscle, and respiratory muscles, and target that genetic basis of disease. Now, our platform consists of three components. In the top left part of the slide, you can see the Fab, or the antigen binding fragment, or the antibody fragment. Again, we call it the Fab. And it's the top portion of this depiction of a full monoclonal antibody, the Y-shaped figure. So we took this top portion of the antibody, the Fab, and designed it to bind tightly to something called the transferrin receptor 1, or TFR1. Well, why is that? TFR1 is highly expressed across the surface of muscle cells, including heart, breathing muscles, and skeletal muscles. So the Fab targets the transferrin receptor 1 and uses it to deliver PMO to muscle while maintaining the ability of TFR1 to transport iron into the cell. At the bottom part of the slide, you can see that we use a clinically validated linker, which we chose because of its demonstrated safety and efficacy in third-party approved products. And this linker enables precise connection of the Fab to the exon skipping PMO. And finally, we have an exon skipping phosphorodiamidate morpholino oligomer, or PMO, which we rationally designed to target the genetic basis of disease. This is where we leverage the modularity of the FORCE platform And what I mean by that is for nuclear targeting diseases such as Duchenne, we choose ASO payloads. And in this case, we chose an exon skipping PMO to target the specific mutation in the DMD gene. And DYNE-251 is our investigational medicine that uses an exon skipping PMO designed to enable skipping of exon 51 to make internally shortened functional dystrophin. And the platform is designed with the goal of addressing the current limitations of delivery to muscle tissue that I highlighted earlier, improving the delivery of PMO to cardiac and skeletal muscle, including the diaphragm. And also, using this technology, we should have increased time between doses. And I'll talk more about this when I talk about the clinical program. Next slide, please. A question that we often get is, how is this different from other approaches? So essentially, we are using the cell's natural biology to deliver our investigational medicine to muscle cells. As I mentioned before, our Fab binds tightly to the TFR1 receptor, and the TFR1 is highly expressed across the surface of muscle cells like skeletal, cardiac, and smooth muscles. So the Fab acts as a kind of key, if you will, that unlocks the TFR1 receptor and allows our PMO to enter skeletal, cardiac, and smooth muscle to target the genetic basis of disease at the level of the nucleus. In a way, we use a Trojan horse approach to drive the PMO into the cell using the TFR1 receptor without disrupting its normal function of transporting iron to cells. So the FORCE platform harnesses the natural mechanism of TFR1 receptor mediated delivery to transport the PMO across the cell membrane. And this is differentiating because we are not using membrane destabilizing agents to deliver the PMO into the cell or to leave the endosome, which some other modalities have to do. By targeting muscle specifically by using TFR1, we have the potential for a wide therapeutic index, resulting in less frequent dosing and chronic administration. Next slide, please. So today, Dyne is well on its way towards achieving the promise of FORCE and delivering for patients. We've delivered exciting clinical data in this program, and I'm pleased to share our latest data in this presentation. Next slide, please. Prior to starting our DYNE-251 clinical development program, we proactively obtained expert feedback. We engaged key opinion leaders, or KOLs, from various disciplines, including pediatric and adult neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy. And importantly, we've gathered this feedback from across the globe. KOLs, both in the US and outside of the US, with experience running early and late stage trials, have provided advice on our overall clinical development program, including our clinical trial design, the patient population that we've selected, biomarker and functional endpoints, and key safety considerations. Importantly, they have provided key learnings from numerous long-term natural history studies to help inform our trial design and eventually to contextualize our results. It's clear that KOLs believe dystrophin continues to be a key endpoint for this condition. We've also received advice directly from patients and caregivers, as well as global advocacy leaders such as PPMD and from the Duchenne Community Advisory Board, comprising of advocacy leaders from several countries around the globe, including Canada. They provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. And this input has been critical in ensuring a patient-centric design. And that advice has included, but is not limited to, key considerations for choosing a clinical trial, review of and input to our informed consent and assent forms, feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the entire experience of participating in our trial. Next slide, please. So before we go into detail on the study design, this is a quick snapshot or overview of the DELIVER study design. Importantly, it's a phase 1/2 global trial that we have designed to be potentially registrational. The trial is enrolling ambulant and non-ambulant males with Duchenne who are ages 4 to 16 and have mutations amenable to exon 51 skipping therapy. Patients who have been previously treated with exon skipping therapy can enroll and deliver after a 12-week washout period. The primary endpoints are safety, tolerability, and change from baseline in dystrophin levels as measured by Western blot. Secondary endpoints include measures of muscle function like NSAA, time function tests, and exon skipping. And we also have wearables to allow collection of data such as stride velocity, which has been accepted by major European regulatory authorities as a primary endpoint with Duchenne trials. For more details on our inclusion-exclusion criteria, you can visit our ct.gov or UDRA CT page for more information. And as you'll note here, we have several sites in Canada, and this is a global trial, as I mentioned. So we have many sites within the US, as well as Europe and Australia. Next slide, please. So this slide goes into a bit more detail on the trial design. As you can see here, we're continuing to step through our cohorts with higher and higher doses. This is the multiple ascending dose portion that's depicted here. Participants are randomized to receive DYNE-251 or placebo every 4 or 8 weeks. We do take 2 muscle biopsies, one at baseline and one at 6 months when the placebo-controlled period ends. After that 24-week placebo-controlled period, all participants receive DYNE-251 after that portion as they enter the long-term extension. I'm pleased to say that although we've fully enrolled our 40 milligram per kilogram dose, and that's the PMO dose when I say 40 milligrams, we have additional 40 mg per kg cohorts enrolling now. And today, I'll be reviewing the most recent efficacy data we reported, and this is at the 10 milligram per kilogram level. Next slide, please. Now, turning to the baseline characteristics for DELIVER, similar to another study, we've designed DELIVER to be potentially registrational to maintain the blind. And so we haven't broken these out by placebo and DYNE-251 treatment groups in order to make sure we maintain the blind. Overall, baseline characteristics are similar across cohorts and generally representative of a population of boys living with DMD amenable to an exon 51 skipping therapy. Next slide, please. Moving on to the safety profile, which we believe is favorable to date for DYNE-251 and based on 48 participants enrolled through the 40 mg per kg cohort. We've not seen any dose-dependent increase in reported adverse events. Most of our AEs were mild to moderate in intensity, and many occurring around the time of infusion. Importantly, no participants have demonstrated persistent related anemia or thrombocytopenia, no kidney injury or clinically meaningful changes in electrolytes, including magnesium, and no withdrawals have occurred either. And with approximately 480 doses administered over 35 patient years of follow-up, we again believe that we have demonstrated a favorable safety profile, which has supported dosing up to 40 milligrams per kilogram. Next slide, please. So I'm going to start to get into the results now that we reported earlier this year. So first, let me orient you to the slide structure, which is the same for the next few slides. The gray bar represents the placebo-treated patients. The blue bars are patients dosed at 5 milligrams, again, PMO dose per kilogram. And the teal is 10 milligrams per kilogram. And you can also see the change from baseline to 6 months. So this slide shows muscle concentration and that DYNE-251 drove dose-dependent delivery of PMO into the muscle. This confirms that the drug is getting into the muscles and more gets in with higher doses as we would expect. Next slide, please. We also saw a dose-dependent increase in exon skipping. Here you can see that 10 mg per kilogram of DYNE-251 resulted in nearly 2% exon skipping. Next slide, please. So we are incredibly enthusiastic about the dystrophin data as measured by Western blot. At the 10 milligram per kilogram dose delivered every 4 weeks, DYNE-251 reached 3.2% normal dystrophin, a change of nearly 3% from baseline. Now, this dystrophin analysis on the left-hand side of the slide is what we call unadjusted for muscle content. And we believe this is an appropriate evaluation of dystrophin quantification. And these data are quite striking, seeing about 3% change after 6 months. However, we know that others in the field have done something called adjustment for muscle content. So we've done that analysis as well. Here you can clearly see on the right-hand side of the slide how we've calculated this, which is essentially the dystrophin, MHC dystrophin divided by percent muscle content. And really the point here is that we're doing a mathematical adjustment based on the amount of actual muscle content in the biopsy selected. DYNE-251 achieved 7.6% dystrophin at 6 months. And this is a change of 6.9% from baseline. We believe this is the highest level of dystrophin reported for an exon skipping therapy for 51 skip amenable participants. And importantly, this is at just 10 milligrams per kilogram. And we're currently dosing at 4 times that level at 40 milligrams per kilogram. Next slide, please. So the 3.2 unadjusted dystrophin expression is nearing the goal of 5 to 10% that we've stated for some time that we believe would drive functional benefit. And what's exciting is that as we near that target level, we see dystrophin production lead to encouraging trends across multiple functional endpoints at just 6 months. So we see stabilization or improvement with 10 milligrams per kilogram of DYNE-251, while participants receiving placebo decline over the same period of time. And we see this on the NSAA, the time to rise from floor, 10-meter walk-run time, and the stride velocity 95th centile. So we see this across various measures of function. We'll continue to evaluate functional impact at higher doses and at longer time points. And while we recognize that these data are relatively small participant numbers right now, we are very encouraged by these data. Next slide, please. So in summary, we believe these DELIVER data reinforce the opportunity for DYNE-251 and more exons in terms of our DMD exon skipping programs. And at 10 milligrams per kilogram dosed every 4 weeks, DYNE-251 showed a compelling profile with 3.2% dystrophin expression. Trends in improvement in functional outcomes, including measures that you're familiar with, like NSAA and time to rise from floor and 10-meter walk-run, and our wearable stride velocity 95C. We've also shown a favorable safety profile to date, and we're currently dosing at 40 milligrams per kilogram. We plan to provide an update on the path to registration by the end of this year. As you know, there are exon skipping therapies that have been approved in the US, and we are committed to developing programs for the globe. We do have confirmation that the pathway remains available to us in the US, and we're having conversations with ex-US regulators, and we look forward to providing an update later this year. We believe this data set from DELIVER illustrate the promise of the FORCE platform in developing next generation exon skipping therapies as well. And we're excited about the opportunity to leverage FORCE to address additional mutations. We do have preclinical programs targeting additional exons, including exons 53, 45, and 44. We all know there's a significant need for therapies targeting these exons and many others as well. Next slide, please. And with that, I would like to again thank Defeat Duchenne for the opportunity to present to you today. And thank you to the families, investigators, and sites that have made this and so many other advancements in Duchenne possible with your countless sacrifices and dedication. What you have all done over the years is nothing short of extraordinary. And so thank you, and thank you for your time and attention today. And with that, I'll invite Catherine to join and take any questions if there are any questions. So I'm looking at the chat and I do not see any questions. Maybe we'll give it just a moment, and if there's no questions, then we will wrap up. And you can see Catherine's contact information here on the slide, and please feel free to reach out to Catherine as well should you have any questions that come to mind later.
Thank you so much. And so exciting. Really enjoyed your presentation. It just sounds like a wonderful opportunity, a great project. And I'm sure that our Duchenne community in Canada will be very, very thrilled to learn about this, and appreciate you offering them the opportunity to reach out with any questions they might have. And I will say the same. If you do have any questions that come up and you'd like to reach out to Defeat Duchenne Canada, you can contact me directly, lisa.mccoy@defeatduchenne.ca, or info@defeatduchenne.ca, and we will definitely forward those questions on to our wonderful colleagues. Dr. Dugar, thank you so much for taking the time. Catherine, Tiffany, I know you're in the background. We really do value. Maybe if I could just ask you to stop sharing your screen. Just want to let everyone on the call know that we are in the throes of confirming all of our plans for our 2024 Family Forum. It's actually taking place in September this year, which is a little bit earlier than we typically hold it. That is to align with Duchenne Awareness Month and a World Duchenne Awareness Day, which is taking place on the 7th of September. So we hope to be able to leverage lots of momentum from those activities as we go in to our Family Forum, and also we'll follow that up with our Advocacy Summit the day after. So if you can join us virtually or in person in Ottawa, please visit our website, defeatduchenne.ca/family forum, and register. And just again, a reminder that if you visit our website, we have lots of resources there for you, lots of information, including emergency care resources, and lots of others. We have community education. You can contact us anytime, but also, like I said, please visit our website. And we now have the community education cards that are available. This is just, if you want to have something to carry around with you to share information about Duchenne muscular dystrophy, and we would be happy to send you those if you are interested. And we have a terrific clinical trial finder tool also that you can access through our website. This tool gives you the opportunity to punch in criteria that will help you to narrow down your search and find clinical trials that are right for you or your loved ones. So we encourage you to visit that as well. And of course, we're very active on social media. So please follow us: Facebook, Twitter, YouTube, LinkedIn, etc. And feel free to use our hashtag #DefeatDuchenne if you're involved in any fundraising activities or education activities that you want to share with the community. And one last thing, if you are not already registered to receive our e-newsletter, we encourage you to do so. You'll receive lots of updates directly into your inbox. So you can sign up for our newsletter, again, at defeatduchenne.ca. And with that, I will just thank everyone again for attending. Thank you to our wonderful colleagues at Dyne Therapeutics, and remind you again that if you have any questions, you may reach out to them or to us, and we will make sure that you get the answers you need as quickly as possible. So thank you all again, and I hope you have a wonderful afternoon.